Hello, my name is Faris. I'm a co-founder and CEO of Tercen, and this is a commercial tutorial for CYTO 2022. So let me start with what if. What if every life science algorithm was simply an app? What if biologists explored and interacted with their data visually? What if analysis pipelines could be shared with just one click? What if reports had dynamic visuals, reproducible calculations, and audit trails embedded? Well, this is available now in Tercen. We had three main communities. We had the biologist, immunologist who wanted to control their own data because they spent quite a lot of time generating it. So they want to control it. They want to explore and interact with it. They want to use the latest algorithms. For example, if they read a Nature paper and they see an algorithm, they want to be able to use that algorithm. They want to easily integrate extra information, especially if it's biological or clinical, and add extra metadata to their data. What does the bioinformatician want? Well, when we talk to them, they say they have all this code that they spent quite a lot of effort making, and they want to make it easily and widely accessible. They want to standardize their processes. They want to automate their pipelines and be able to scale to high level of a large number of data sets or low level of data sets. And they want effectively to have a better support the biologists and the immunologists. And better support means in terms of higher value support, but also the number of biologists, immunologists they want to support. And what do the IT community want, especially the departments in large pharma or even in biotech and in universities? They want a high level of data security, especially now with the cloud technologies. They want to control their computing resources in terms of CPU and hard disk. They want to elastically be able to respond and increase the number of CPUs and hard disk or decrease it depending on the demand. And they want a flexible licensing model that can match this flexibility in number of users and resources. So Tercen aimed to do all this after discussing it with them, and Tercen had to innovate. And one of the things that we innovated was this interactive system. The system had to allow immunologists or researchers to see their data, because if they see their data, they understand their data. And this is what we call offloading cognition to perception. The system, to do this, the system had to handle billions of data. The system has to be flexible on the experimental question. It has to be easy to add extra information. It equates visualization and computation. So what you see is what you compute. It has to interact at the speed of thought, and it has to be simple. And this is what we did in Tercen. But we also had to add another innovation, which was take any code that's there in GitHub or in the repo, and especially in this rich open source ecosystem, and take this code and wrap it into an app, so as the immunologist can use it very easily. And so we had to create this abstraction layer to do this, and we can now go from code to app pretty quickly. How do we achieve all these innovations? Well, it's basically because we have these three layers in Tercen. We have the statistical layer, where the open source code basically resides as an app. We have a relation layer, which is all about managing the relationships between the different data, especially when you come from raw measurements and you do primary, secondary, or tertiary analysis. You keep the connection between all three. And then we have this highly interactive system at the very top where you interact with your data. In all three layers, we have patents pending. And we're pretty excited about the architecture. And I've selected three demos to show it to you. The first demo is a high parameter data set, and it shows the flexibility of Tercen in handling high parameter data. The second is to show clustering across a time point or clustering across disease severity, and that's to show you how Tercen is flexible with the visual aspects. And the last one is showing Tercen how we can integrate, where we're integrating T cell receptor flow data with genomic data. Hopefully you'll enjoy the demo. Okay, let's look at one of the projects. I've chosen here to demo a Omics 69. It's basically an Aurora Cytec 40 color data set. And it is indicative of what we can do with Tercen. I click on it and we enter the project, and you see there's quite a lot of files here that are composed of our analysis, and there is a README below here. The README is a text and has dynamic visuals embedded in it. It's a way of blogging or communicating to the reader what was done on the analysis. All these images are dynamic, they're embedded, they're pointing to the workflows that you see up here. We have not just workflows, but we also have files and data files. So let's look at one of them. Let's click on one of the pipelines. Here it is. This is a typical Tercen pipeline. It's composed of steps, as you see here, all starting at the start with the FCS files, joining with some meta annotation, pre-processing, performing flow sum here with n equal 40 clusters, performing a UMAP, performing a TSNE, doing a Mem score, and then doing some automated population annotation. Okay, let's look at the TSNE plots. If you double click on the TSNE step, you'll see that you have 4 views. One of them is a view where you can see the TSNE plot with TSNE1 versus TSNE2 colored by the clustering. And in this case, it was the flow sum clustering. So you're seeing a TSNE plot here with 40 clusters. Okay, let's look at another view. If I double click again on the TSNE, I can select a different visual. This time is TSNE1 versus TSNE2, but be colored by the expression level of the marker. I would call it the absolute scaling, but we also have a similar view if we double click and we take the scale view, which is again colored by the expression of the marker, but this time each marker is scaled. So you get a better overview in terms of relative scale. Okay, let's look at another step, the Mem Score step. A Mem score is an enrichment calculation. It comes from the Jonathan Irish lab. But if I double click on the Mem score, you will see what it indicates. Each column here is a cluster. There's 40 clusters as indicated by the flow sum that had previously been run. And each row here is a marker. So you're getting basically an enrichment score for each marker for each cluster. And the enrichment score, which is indicated here by the color, is a plus 10 or a minus 10 or anything between. A plus 10 means it's enriched, a minus 10 means it's depleted, and white is basically neither enriched or depleted. So it's a good way of having an overview of what is enriched, which cluster is enriched with what marker. Okay, after the Mem score, we have the automated population annotation. This is based on trying to give names to the clusters that have been identified with the flow sum and enriched with the Mem score. So if I double click on it, and you will see what I mean here, this projection, you see the immunophenotype groups on the rows, and there are the clusters, or the 40 clusters on the columns. And in the first entry here, you see B cell. You can see that B cell is composed of multiple clusters. And this is based on an annotation that has an algorithm that has gone through each cluster and given a immunophenotype naming to it in such a way now that it makes more sense and starts indicating to the scientists which clusters belong to what immunophenotype. All right, I'd like to show you the input file for this algorithm of automatically annotating clusters with their population names. And this is the file. It's a simple Excel file where every row is a population name and every column is a marker. And the actual entry here, like 1, 0, or -1, indicates whether for example, in this case, T cell is enriched or has to be enriched with CD3, and it has to be depleted in CD19. And 0 means it's not important. So in this case, CD56 is not important for the T cell definition. And the immunologist fills out such a file and uploads it to Tercen, and the algorithm will use this information to go through each cluster. In our case, there was 40 clusters, and give a population name to each of the clusters based on this definition here. Okay, so I'd like to show you how interactive Tercen is in terms of building up these visuals and computations. I've prepared a totally empty data step at the very bottom, so as I show you how to build it up, let's double click on the empty data step, and it opens up and shows you on the left here all the factors that are computed in the data analysis workflow. And you'll recognize the workflow on the left, going from the export to the joining of the metadata to the pre-processing, the flow sum, UMAP, TISNI, Mem Score, and automated annotation. So let's build up something. So I'll do a TISNI plot effectively. So I will click on the TISNI step here, take TISNI 1 and put it on the x-axis, and take TISNI 2 and put it on the y-axis. And I'm going to color by the flow sum clusters. In this case, it's called meta cluster ID. So I drag it over and I drop it in the color area. So this is a TISNI plot colored by a flow sum color. All right, let's say I want to add extra information to this visual, perhaps bring some donor information. I'll go over here to the join metadata step, and I see there's a factor here called donor. This came in with the metadata, and there's 4 donors in this data. So I'll just grab donor, pull it over, and put it on the column. It'll split the TISNI plot into the respective donor contributions. This is a particularly useful view if you're looking for donor-donor differences. Now I can go a little step further and take, for example, maybe the cluster, the flow sum cluster, the meta cluster ID in this case, and drag it over and drop it on top of the donor. This gives me a view where the TISNI plots have been split up into their clusters, but also into the different donors. So it's a per cluster, per donor view, and seeing how each of the 40 clusters in this case differ across the donors. This is useful again for looking at donor-donor differences, but let's use the annotation population information, which is at the very bottom here, the automated annotation. And you can see here there's quite a lot of populations that have been identified, but I'll use this factor here and I'll drop it as a filter, and I say I'll only want to look, for example, at the T cells. I want to focus on the clusters, so I click on T cells and I say OK. So this will activate a filter that only selects T cells. Now we're looking at the T cell populations, and notice that there are multiple clusters per T cell here, across the different donors. So this filtering step here allows you to zoom in to the particular population or particular donor or particular cluster, and a very powerful feature here to interact. So you can build up quite powerful and informative, insightful type of views here. All right, that was the tour of the powerful visual features of Tercen, and especially useful when you're applying it to high parameter data. This workflow can be saved up here and we can progress to the next project. All right, let me show you another project. TrackSum it's called here, and it highlights the TrackSum algorithm. TrackSum is an algorithm that allows you to cluster across time points of disease severity. Here I've clicked on it. Here are the data sets in here, as well as the pipelines and the README. The README in this case highlights the different data sets and the publication that outlined and explained the TrackSum algorithm. So let's look at one of the data sets. It's a toy synthetic data set. I'll just click on it. And this is the pipeline used to process the data set. It's effectively loading it up, adding some metadata, just like the different time points, so day 0, day 1, day 2 time points. And a pre-processing is performed, TrackSum is performed, TISNI is performed. And let me just look at the data set first. This is a TISNI plot of the data set, and it's been colored by the populations. In this case, there's been 11 population groups, and it's the ground truth or the absolute truth. These are the populations that in day 0, for example, you see you have population 1 and 2, and it progressively splits into more refined population groups. It's a synthetic data set to test the TrackSum algorithm. What you're ideally looking for is an algorithm that can detect how these population groups are changing through time from day 0 to day 4. And so let's look how TrackSum performed on this data. And if I open it here, what you have in TrackSum is that It found clusters, and in this case, it's the rows that are called the clusters here, and it gave them these letters A, B, C. It found a cluster A, which basically captured population 1 across the different time points, and it was able to detect or track the fact that day 3 and day 4, the actual population changed, but it was still the same cluster of cells. And you see in, for example, metacluster B here, it detected the big cluster 2, how it also fragmented into smaller clusters here, and it was able to successively keep the information of these cells and how they change through time. So Traxome is quite useful for tracking the cells through time. It's quite a powerful tool. All right, I'd like to show you a last project. It's a T-cell receptor project. As you open the project, you see all the pipelines, the data, and the README. It's a simple little README. It just talks about the patients, how these cells have been sorted using 4 channels, using a Sunny SH800 sorter. And once they were sorted, the cells were sent to the genomics core lab, where they got sequenced with the MiSeq instrument. And these data sets, or this data that was generated, needed to be combined, and Tercen was used. And I'll just show you the pipeline. It's a very simple pipeline. We have the fact sorting here coming from the sorter. We have the plate annotation that came from the sorting, and I'm joining them here to join the 2 informations. And then we have the sorted cells that are sent to the sequencing machine, and the sequencing results are coming back. Of course, they're connected with the well or the plate ID, and you join them here with a joining on the well ID or plate ID. And then you perform, because they were sorted with these 2 extra channels that measure the epitope binding strength. Here, the 2 epitope channels were pre-processed, and then this view here, which projects on the X and Y the 2 epitope binding strengths on the T-cell receptor, and it's colored by the sequencing information that's coming from the genomics lab. So you're combining 2 sorts of data, the facts sorting data plus the genomic data here in one view. And from this view, the scientists can see if there's a strong epitope binding, what could be associated, what is the T-cell receptor genomic sequence related to that strong binding on one epitope or on the other epitope or both at the same time. Well, I hope that was clear for you on all the types of projects, but we have much more. So to recap a little bit, we can basically take any algorithm, and you saw Traxome was one of those, and we can present it easily to the biologist. We can package full pipelines, and you've seen a few of them already, and they can be shared and cloned. I hope you've seen also the interactive visualization, where I could manipulate in quite innovative ways and project anything I would like from the data. And I've shown you some of the README, where you can see how Tercen is very linkable, and every view you have seen has a URL that can be shared. We also have a very flexible way of deploying, either on local machines or cloud. Thanks very much for listening, and I'd like to thank the whole team. Hope to see you at our booth 245.